Thanks, Dr. Cantor, and I would like to thank all of the speakers for staying on time, which now gives us almost the entire 30 minutes for the discussion. If anybody has any questions, please feel free to come up to the microphone. This was a lot of food for thought.
Martha Summerman, NIDCR. I had a question about the vision, and this is more of a longer-term theme of functional vision versus visional function. And as an ophthalmologist, you probably know we still use the eye charts for determining vision, but that's not the functional vision of a patient. And is anyone going to change that someday?
So what I would say about that is it's an extremely informative test that, central 4 degrees of your visual field, but clearly that doesn't define your entire visual experience. So there are other tests that we use, in a clinical sense. But if we were going to be doing and have done other gene therapy trials for inherited diseases, it's a new concept, I think, for some regulatory agencies, but we're pushing visual acuity away, the eye chart vision away for other type of testing for the reasons you state. It becomes irrelevant for certain diseases.
I have a question for Dwight about Pompe. You mentioned that, I guess for adults who are on enzyme replacement, that the stability of their findings is such that if they see an improvement after gene therapy on either, was it FVC or 6-minute walk test, that must be attributed to the gene therapy intervention? Do you think that's a generally accepted observation?
Well, I mean, there's a lot of data on the long-term trajectory for patients who are treated with ERT. And the data cumulatively say that there's initially some improvement in the 6-minute walk test and stabilization of the FVC of the first couple of years, and then a gradual decline. So I think it's fair to say with large groups, this is the data, large groups that we understand their trajectory on ERT and when we can expect to see improvement. But talking about individuals, it'd be very, very important in looking at their own baseline and their history. And I wouldn't say, I wouldn't say that any improvement in any individual who's been treated for ERT a long time could be attributable to this new intervention. There's so many variables, but I think that an adequately powered study with appropriate controls and referencing the available data could be used to support benefit of a new intervention. And of course, the gold standard or maybe the most reliable data would be to treat naive patients who've never been treated with ERT, but they're very, very hard to find, especially in today's environment.
Hi there. Paul Mellmeyer with the Muscular Dystrophy Association. I also have a question for Dr. Koberl. On your slides, you cited a handful of endpoints that are used within Pompe disease. Can you speak to the patient community's viewpoint of these endpoints, if they reflect what the community is seeking within new innovative treatments for the disease? I know within other neuromuscular diseases, the 6-minute walk test isn't particularly popular. Is that true within Pompe as well? And I guess a follow-on to that is, are there any efforts ongoing to develop patient-focused, patient-derived endpoints that do reflect what they're hoping for with the new innovative treatments?
Well, yeah, and I think there are better endpoints than the 6-minute walk test that are more clinically relevant, some that we have available already. And so there's the Gates-Stairs-Gower chair, which the name implies measures more important functions that are relevant to daily life. And so those endpoints are validated in that they've been correlated with 6-minute walk tests and with clinical condition in Pompe disease, and they'll probably be used going forward. But in terms of developing newer endpoints, we're really looking at what's already available. I think that'd be a really important creative process to come up with a more disease-specific and appropriate endpoint. But even there are even publications. Benedict Schoscher wrote one. 6-minute walk test is that all there is? So clearly, I think we need to look more closely.
Hi, Rachel Salzman. I'm a member of the Forum of Regenerative Medicine. I think my question is for Larissa. I really appreciated your introduction. I think what's unique about gene therapy is that we know what we're targeting, right? Especially if you've got a loss of function. So can you just make a comment about how we should think about the biomarker that we have delivered the gene and the gene product is present? Because we know if the gene is intact, you don't have the disease, and if the gene is mutated, there's, obviously that downstream lesion. So if you're able to demonstrate as maybe an early, biomarker of target engagement that you're getting the gene product, what's the utility of that, or how to think about that on the pathway?
Well, that's a great question, and not only that it talks about an important concept, it also is the question that comes up so frequently. We see that here is the gene therapy, and here is the transgene, and here's the transgene protein that's there, but what does it do? Right. So under the current regulatory framework, we're looking, and this is really something that was said a number of times. What we see has to be clinically meaningful in order to be used potentially as a measure in clinical trials. And it is this step between seeing something in the laboratory and making sure that this something that we're seeing actually translates into actual improvement in patient symptoms. That's often a step that requires a lot of deliberative thinking and a lot of work and a lot of data understanding and analysis.
I think there's an opportunity for us to leverage this sort of common gap.
Right. Because remember, we're always looking at how a person feels, functions, or survives, because that's the most important piece.
Hi, Mildred Cho from the Stanford Center for Biomedical Ethics. I have two questions for Dr. Cantor. Thanks for your excellent presentation. First, I'm stunned to hear that there is no registry for sickle cell disease. So, okay, maybe, but so just your 2 cents on that. And then also, you didn't really have time to talk about the stopping points issue, but I'd like to know sort of your take on how people are thinking about that now.
Absolutely, thank you. So at this moment, there is no national registry. There are several different groups starting to put together different forms of registry, and obviously collaboration is going to be key in that we want to make sure that too many people aren't in the kitchen cooking up different registries. It really comes down to a lack of together advocacy, a lack of funding. We've had in the sickle cell world, no strong funding body that's really worked to our benefit to advocate for this on our behalf. In recent years, the American Society of Hematology has undertaken sickle cell as their first disease-specific initiative, which is making a great difference. And NIH and NHLBI have certainly embraced sickle cell disease as well. However, have said long-term that they're not interested in being the long-term funder of a registry. So that's really impacted our ability to sustain one. On the second note, stopping rules. So right now, there's no firm endpoint guideline or stopping guideline in these trials. So I think Derek Robinson said earlier, something that's very important is that all gene therapy is not created equal. And I've actually devised some education material to explain the differences and what patients need to start asking, because I think it's important that when they go into a gene therapy program that they realize that gene therapy A may not equal B, and A may be disease modifying and B may have the potential to be more curative. So as we gather more information, I think we'll have the ability to say that if you're looking, for example, at a gene addition therapy in which you see very low and persistently low vector copy numbers, we're going to know that that's going to mean low protein production and insufficient hemoglobin production to be disease modifying. But at some point, because there are so many unique trials ongoing, we're going to have some form of stopping rule that's outside of just safety but also efficacy, because these are myeloablative transplants.
Hi, John Tisdale. I wanted to get back to the prior question and push Larissa just a little bit more. So we have these, requirements in sickle cell disease to have some change in their clinical state, which is sometimes difficult to measure. We've heard from Julie how difficult that can be. But I wonder when we're going to get to the point where resolution of the anemia is an endpoint, just like resolution of hypertension is now for drugs that are designed for hypertension. So in the beginning with hydrochlorothiazide, it had to be proven that a course of an antihypertensive would in fact reduce the risk of stroke in that population. But now we're comfortable knowing that reduction of blood pressure does reduce that risk, and having a blood pressure medicine only reduce blood pressure, not the risk of stroke. So I wonder when it is we're going to cross that threshold for a disease like sickle cell anemia, where resolution of the anemia is an endpoint that we can use in a clinical trial.
So I think it's important, to think about how a particular measure really reduces the disease burden, right? Because if you're talking about improving hypertension and then eventually preventing strokes, you would want to think about how improvement in the anemia, for example, and I'm not wearing my regulatory hat now. I'm wearing my researcher clinician hat now. You would want to think about how it reduces the disease burden. Does it lead to fewer hospitalizations? Does it lead to improvement in respiratory function? Does it lead to less fatigue in that anemic patient who is not anemic anymore? So there are always ways to look at the reduction of the disease burden from the perspective of how you improve the symptoms. And then if you start thinking about it, from that perspective, then there are ways to potentially look and validate the endpoint that you're talking about towards how it improves the outcomes in a patient.
So in that light, and I apologize for interrupting, I think the question, though, translates to in stem cell transplant, in allogeneic stem cell transplant, we know that the protein production, hemoglobin A, it may take 2 to 3 years to make that pain go away, because that is what happens on sickle cell disease. So I think the concern is that, and it may take that long for fatigue to improve and organ function to improve. And I think there's concern that when we know that we're producing hemoglobin A, that acts, walks, talks, and is hemoglobin A, that it will equal the same thing. It's not a question that we're making a new hemoglobin. It's the same protein just being produced with a transgene. So I think the concern is, do we have to wait 5 years, 10 years, because it could take that long to see some of those downstream clinical manifestations. And some stem cell transplant participants in sickle cell disease will have pain for 3 years post-transplant due to various comorbidities and opioid dependence, as well as bone infarct. So I think we worry in the community that translation could take way too long to reach individuals with sickle cell.
Jay Siegel, I have a question for Dr. McGuire. I understand that in recent years there's been a lot of work on a technique called microperimetry, which at least theoretically has the ability to repeatedly over time interrogate the same point on the retina, as I understand it, for its sensitivity to variable levels of light. And that sounds to me, at least theoretically, as to have the potential to be a sensitive measure of retinal disease over time. What's the status of that, and how are there data comparing that to multiluminosity mobility testing?
So I would sort of give the analogy of pupillometry, extraordinarily sensitive test, and it's a new technology, reproducible, and we use it. But again, we're thinking of it as a secondary endpoint because there is no strong correlation that anybody has done with walking through a town or with occupational therapy type evaluations or self-reported VFQs. So again, it's one of these wonderful tests of visual function, extremely precise, but doesn't yet correlate to that clinically mean.
Are you potentially working on that sort of validation of correlation?
I think eventually everybody is trying to do it. It's similar to looking at blood pressure and stroke. Yeah, absolutely.
I have a question for all of the speakers. We still have some time until 3 o'clock. I have to be a little provocative, particularly, in light of the earlier sentiment about longer term and, more observations. So there are opinions that it may be difficult, if not impossible, to validate a surrogate endpoint in a rare disease. You have very few observations. Time is pressing. So could you tell whether you would agree that it is not possible to validate a surrogate endpoint in a rare disease, or whether it is possible, and if you could discuss a little?
I think I have the same problem that they're discussing, in that a lot of the diseases we deal with, the timeline is so long that for people to maintain their attention and funding for 5 to 10 years, it's going to be very difficult. And that's in order to even develop a surrogate measure. I think that's a problem that probably faces all of us.
I think it is possible in sickle cell disease. Certainly one of our biggest problems is this lack of a registry. We would have a much better understanding of what in a 2-year-old would predict mortality in a 25-year-old if we had a longitudinal registry. So I think it is possible. I think we need additional sort of universal funding to get that together to ensure that we can answer some of those questions. And I think we're beginning to see more of those answers. So I think it's very possible. I just think we haven't had the right tools or the right people at the table until recently.
Yeah, I may be too optimistic, but I think we probably have a couple of candidate surrogate endpoints, at least in the rating scale that you provided, and it's just a matter of time collecting data and then the review process to really confirm validation.
Great, and we have some more questions from the floor.
So this is a follow-up to what you actually were talking about here. So endpoints, this is a great conversation, and endpoints are so critical. And yet, as you mentioned, it's easy to do a test that on the eye will take our vision, that's simple, quick. So how these endpoints are going to take longer, perhaps? And I just wonder how you predict, or anybody in the audience, and the cost of it, and how are we going to do it, not just for rare diseases, but I think we need better endpoints for a variety of things we're doing, and how we're going to cover this.
Not to directly answer your question, but I would also say that one of the issues from an academic point of view, if I'm a young investigator trying to get promotion or funding, endpoints is not where I'm going to go. I'm tenured, so I don't really have that issue, and my wife and I work together, and she forces me to do this, so I have to do it. But I think one of the real fundamental problems is how do you maintain the long-term picture and encourage people to fund and to look at endpoint development of endpoints and endpoint research as something worthy of an academic endeavor.
I think we get lucky in sickle cell disease. Endpoints is really a hot topic. So I actually know several junior researchers who are following this up. I think it's critical. I have a close colleague who just actually wrote an R01 specifically trying to design a new measure of GFR kidney function, because the current GFR is not accurate in sickle cell disease at all and cannot be used at all in a validated trial. And so I think that there's great need there, but I concur with many of the concerns in academia, which is the increased push for RVUs and not research. And so it has to be up to the various organizations to realize the importance of those endpoints in order for us to persist in research.
One comment as a reviewer, just a sec, but I think that the availability of potential new therapeutics make it much easier to justify and develop excitement for your proposal. But if the therapeutic is not there and it's a rare disease, I think that's an incredible uphill struggle.
Yes, please.
Yeah, hi there. Todd Durham, Foundation Fighting Blindness. The question I have is related to the previous, which is we do spend quite a bit of time wanting and trying to develop new outcome measures. Companies come to us and say, how can we get regulatory feedback on these novel outcome measures outside of a development program?
So that's the question? Mm-hmm. Okay, well, that's a great question because I know an answer to this question. So we have a program that's called Drug Development Tool Qualification Program, through which one or a number of people, a number of stakeholders rather, could qualify a measure. And those measures could be clinical outcomes assessments or biomarkers. And it's typically, or at least the program is intended for it to be done in pre-competitive space when it is safe to have conversations without the worries of disclosing proprietary information or the worries of competition. So in pre-competitive space, to have these discussions between those who would like to develop a measure and the regulatory agency. If you did not know the answer to this question, that's a bad job on our part, and I will be happy to provide you with a link to the online information about this program.
My question is more to Larissa, and it's more of a kind of regulatory question. So most of this conversation has been in the context, I gather, of getting new gene therapies approved, but there's this whole post-approval process. And my understanding is, at least in cancer, when you have accelerated approval, FDA usually will condition approval on the launch of some kind of a confirmatory study that's using a clinical endpoint like survival instead of, say, response rates, which is what's getting the approval. And there's a large literature now about some of the lengthy delays between the conditional approval or the accelerated approval and the completion of those studies that have clinical endpoints. I don't know as much about what the literature says. First of all, I don't know whether that requirement for conducting those post-approval studies obtains also in the context of orphan diseases, and if so, what we know about the promptness of the follow-up or completion of those long-term studies, granting, of course, that there may be good, reasons having to do with the disease for that lengthy delay. Do you understand the question?
Yes, I think so. So first of all, just to make a few clarifications, because I recognize that when you were asking, you may not have had all the information. So any product, it doesn't matter whether it's cancer or non-cancer, for any indication, right, any condition. So this pathway, the accelerated approval pathway, is applicable to all conditions. When a product is approved under this pathway, it's approved based on some evidence that was generated with endpoints that reasonably likely predicted clinical benefit. Now, the clinical benefit has to be described and verified, and this is done through this requirement to conduct a post-marketing study, which would be typically a clinical trial with clinical endpoints as measures in that trial. Now, what happens in practice oftentimes is that trial is typically already started at the time when the approval happens, or at least it is in the plans, so the design is very clear. It is extremely rare, if ever, that the study would be done, sometime down the road. Typically, the plans for that verification are already in place at the time of approval. Now, how long does it take? It depends. It depends on the disease, it depends on the endpoint, it depends on the clinical benefit. The other aspect that you may have been, alluding to but didn't ask directly is whether, are you FDA a paper tiger? Can you do something about this, right? Or can you not do anything about it? So this requirement is conditioned potentially on the ability to take the product off the market. Sometimes it's done, and there are examples in life when the verification didn't work and the clinical benefit hasn't been demonstrated in that post-marketing study. And sometimes companies take it, and sometimes we have to do it. There are examples. If we have to do it and there is resistance or disagreement, we have to take it to advisory committee meetings. We have to receive advice from experts to see what different stakeholders as well as consumers think about it. But I'm sure you know the stories of bevacizumab and the story of olaratumab and the recent story of McKenna. So, but these are all examples when the product that was looking very promising in the pre-marketing program was approved based on surrogate endpoint, did not end up working well for patients, and so in that case, we have to take action.
Well, it's 3:00, so I could wait.
It's okay. We can take your question.
I just wanted to make one comment, which is, to me, gene therapy will not reach its full potential if it doesn't tackle diseases that have never had treatments. And to the extent that it does that, there will be the necessity to develop novel endpoints. And honestly, as Dr. McGuire says, so I'm glad for Dr. Cantor that there are people who are willing to work on novel endpoints for sickle cell, but to develop a novel endpoint actually was a fairly substantial undertaking, and it was also fairly expensive. I mean, we really did not have the resources to work on that separate study to establish the performance characteristics until we were in the company. So we were definitely building the airplane while we were trying to fly it, because we had to do that study to really look at the performance characteristics essentially after we were already planning the trial to use it. So I think this is a very important panel, but I think we should really not underestimate the challenges involved in coming up with new endpoints that are really accepted by the clinical community, by regulators, and so forth. But it's really an important part of gene therapy, I think.
Thank you, and that's the best conclusion that I only wanted to wish for. Thank you. Thanks, everyone. So we're on break now.